By the way, I'd like to just sort of do a little bit of a tangent today and talk about the blob. And this is sort of a little side thing I've been discussing with my students for the past couple semesters because I think it's particularly interesting. And it's not the blob like, the movie here. It's the blob like this. And what is this blob? Well, you might have seen this article, you might have seen this news popping up in late 2019 about the blob, a smart yet brainless organism fit for sci-fi, gets its own exhibit at the Paris Museum. And this is what it looks like growing in nature, and it's a pretty cool organism. So I'd like to just take a few minutes to talk about it because I think it really nicely relates to our very first class when we sort of discussed what is size for microbes, what does it mean to be differently sized from microbes, and what is really considered a microscopic organism. So the organism, the blob, is Fusarium polycephalum. It's the many-headed slime. So this organism is a slime mold. And you might ask yourself, what the heck is a slime mold? Because, to be honest with you, fungi and animals. So it's sort of this hybrid. And so it is technically grouped as a protist. And a protist is any eukaryotic things where they can assemble. So if you have two slime molds that meet one another, they can actually fuse together to act as a single organism. Like fungi, they do reproduce with spores, and they are useful for decomposition. And historically speaking, they were actually classified as a fungi. But unlike fungi and unlike many microscopic organisms, they move. They also lack chitin, which is a primary common. If you go to a nice now you might think to yourself, why the heck does this unremarkable thing on display at a zoo that has tigers, lions, giraffes, rhinos, right? Big, cool looking organisms. Well, it opened this past October, and it is actually a relatively common gentless bob. So why does it get this fanfare? Because it can actually do some pretty amazing things and some things that are actually kind of out of sci-fi. So let's talk about what it does. So they can repair themselves. And what do I mean by repair? Well, if you cut a piece of the slime mold, so say we're looking at this piece of slime mold here, if I cut this right here, where my mouse is going over, it will essentially repair itself. So it's self-healing. The interesting thing is you can also the interesting about this is the speed. So it takes just a few minutes for it to do it. And it's kind of interesting that if you cut one in half, the halves will move back to one another. And so just put this in perspective of what's happening. Pretend you chop your arm off and you throw it across your bedroom. What if your arm was like about a Fusarium? What it does is your arm would crawl back to you, climb up your body, and then reattach to you the exact same way it was. So again, really out of science fiction for what these critters can do. They can learn. And so as you can see from this picture right here at the top, they are able to solve mazes. In addition, if you challenge them with obstacles, they can actually learn and retain that knowledge. And so you might be thinking, well, what does that exactly mean? So for instance, if I have this slime mold, it's growing on a petri dish. If I challenge it with something that's toxic, such as caffeine, which is a pretty toxic compound, it will sense that the caffeine's toxic, learn that the caffeine's toxic, and then avoid the caffeine just so it doesn't kill itself. Now, if it's ever re-exposed to caffeine, it knows right away, without any learning, that caffeine is bad, and thus it should stay away from it. And so this is something that we didn't really think unicellular organisms could do. It's really something you think about like an intelligent animal could do, like a dog or a cat or even a human. And the sort of interesting thing about them is they can also share knowledge. And so these slime molds can fuse together and merge, as I mentioned. And the sort of interesting thing is once you merge two slime molds, so say if I have a slime mold that's habituated to an obstacle such as caffeine, and it emerges with another organism that has not been habituated to caffeine, the entire organism will now have the collective knowledge of both of those organisms, and thus even the part that used to be part of organism 2 that was not knowledgeable at caffeine will then become knowledgeable about caffeine. So they have the capacity to share knowledge. And it's sort of interesting is it doesn't really matter how many you fuse together. You can fuse 10,000 of these things together. They'll still behave like a single cell. In addition, these guys have some pretty wacky sexes. So there's some articles that put the number of sexes up to 720 species, which is in contrast to the sort of the two classical biological sexes of most animals, right? Male, female. Um, and so these organisms, they reproduce by spores. And so this is what it looks like when a Fusarium is starting to sporulate. It produces these little fruiting bodies to release its spores. You could think of spores, almost like, sperm and egg. But once they reach sexual maturity, the slime mold actually carries two copies of a gene, and each one of those genes can come in a variety of slightly different types. So they can vary at the genetic level, and subtle variations in these two genes that determine sex in Fusarium can lead to different sexes. So it's not like humans where it's XY or it's some even something more complex like birds where it's XXX stuff like that. This individual, it's just subtle changes in genes, and subtle changes in genes confer a different sex. And so if you sort of just look at all the genetic variations in these genes, it leads you to make the number about 720 different sexes of this Fusarium. So pretty crazy stuff. But the sort of the last really interesting thing Fusarium does is they can solve complex problems that takes engineers and supercomputers such as city planning. So there's a really awesome experiment. If you're interested in it, I'd be happy to send you the article, but it's called the Japan Railway Experiment. Now, for those of you that don't know, the Tokyo metropolitan area has a pretty robust railway system, and it took engineers decades to design this railway system because they wanted to maximize it for efficiency because that saves money. So to sort of get at this idea with Fusarium, what they did is they took Fusarium and they put it on a petri dish, and they put little oats, which the Fusarium likes to eat, and they raid those oats just like the main cities are in the Tokyo metropolitan area. What they did is they let the Fusarium grow and link to the oats with connections. So you can sort of see how it's growing. It starts, it expands out, keeps expanding, getting to the oats. By the time it makes it to D, it's finally made, sort of almost all the oats. And then by the time it hits E, after 16 hours, it's made it to all the oats. And after 26 hours, the experiment is concluded. But what you'll notice is that it sort of formed a connection between all the oats. And it did this to maximize its own efficiency, to minimize how much waste it has, and to minimize overall how little of a time it should take it to do it. And it's kind of interesting when you overlay Fusarium over the Tokyo metropolitan area, it actually matches up pretty well. So the network made by the slime here in C is actually strikingly similar to the Tokyo railway system here in D. It has subtle variations, but again, just notice how similar these two pictures are. And that's what's really remarkable, right? This thing doesn't have a brain. It's just a single cell. And it managed to solve a problem of connecting, about a couple dozen oats or cities in a very short period of time without any meaningful thinking, all just trying to maximize its own cellular efficiency. And again, this is something they do with no brain. They have no neurons, no tissues, no multiple cells. This is a single cell cellular organism. And again, it's essentially a gigantic bag of nuclei. This organism can have hundreds of nuclei in a single cell. So pretty interesting little, just sort of little tangent about a really fascinating microorganism. Not someone we're really going to talk about, but I like really interesting cases like this because it shows and it really blurs the line between multicellularity and you know and unicellularity and what microbes can and can't do. And the last sort of thing before we move on to the topic for the day, and is I realized I missed this in my previous lecture, and the question I hope you guys were all thinking, maybe you weren't, but maybe you were thinking it, is are all cells in a culture in the same growth phase? And this answer to this question is twofold. The first is it depends on the media. So in a well-mixed broth of culture, so a liquid culture, the answer to this is yes. But when you look at a colony growing on a plate, the answer is actually no. And so this is a really interesting look at a Pseudomonas biofilm. For those of you that have never heard of a biofilm, we're going to be covering them pretty in depth later in the semester. But just sort of jump the gun on what a biofilm is. It's just a group of bacteria growing on top of one another. Now, the best example I can give you what a biofilm looks like in your average everyday life is if you don't brush your teeth. Say you fall asleep, you forgot to brush your teeth, and you forgot to brush your teeth in the morning. And then by midday, you got this sort of grimy, grossy stuff all over your teeth. That's a biofilm of the bacteria in your mouth. So that's what a biofilm is, just bacteria stacked upon one another. And so what we're looking at here is a staining study where they looked at how, in terms of what growth stage our Pseudomonas aeruginosa culture was in. So green cells are in log phase, so they're expanding their population. But the red cells here are actually in stationary phase. And so when you have a colony, when you have a biofilm, you have very differing levels of growth, right? With the ones that are by the surface, where there's oxygen, they're growing rapidly, they're in logarithmic phase. Those that are sort of below the surface, they're in stationary phase because they lack oxygen. Now, Pseudomonas aeruginosa is a facultative anaerobe, meaning it can grow with or without oxygen. So these organisms are potentially still growing and dividing, but they're not doing it the way they would do it in logarithmic phase. So let's move on to the new material for the day, and this is going to be microbial genomes. And so let's take a brief moment to review bacterial cell organization. So bacteria, very, very simple, remember. They have a envelope and a cell wall and maybe some other structure there as well. They have ribosomes and they have the cytoplasm. So this is all that sort of purple gunk here. But you notice they have this bright red region here, and they have a couple ones over here, but we're just going to focus on this one. This is what we call the nucleoid region, and this is where the bacteria keeps its chromosome. Now, this is in contrast to our eukaryotic, so us, plants, fungi, things like that, that have a nucleus where they keep all their chromosomes. Our bacteria do not have that. Remember, our bacteria lack organelles. And so the chromosome of a bacteria is very, very simple. They are double-stranded, like all DNA is. It is circular. It can be linear, but it is typically circular, and it's typically just one chromosome. And this chromosome is relatively small. It's a few million bases, and this is, I'll just give you an example. This is in direct contrast to a eukaryotic chromosome. Majority of the bacteria's genetic material is stuffed in this nucleoid region. Now, let's look at the two components of a bacterial genome. So this is just a really handy table. So the core gene pool is comprised of DNA elements. So these are the chromosomes and potentially some plasmids. And the features that are encoded on these core genes included ribosomes, cell envelopes or cell membranes or cell walls, key metabolic pathways, DNA replication, as well as nucleotide turnover. Now, that's what every bacteria needs. That's also what every eukaryote needs. That's what you need as well. But there's sort of what we call a flexible gene pool, where there's other DNA elements that incorporate other potentially valuable pieces of genetic information. And these include genomic islands, which typically encode for pathogenicity, so making bacteria better pathogens. We have genomic inslets, which typically confer antibiotic resistance. There's also pieces called as part of bacteriophages, and bacteriophages are viruses that just infect bacteria, and these are typically involved in secretion. We have plasmids, which also, again, are involved with many things, but one of the things they're most commonly um, encode for symbiosis. And then we have integrons, which is typically relying on degradation. And then finally, we have transposons or transposable elements, which typically include secondary metabolism, modification, how they chop up their DNA, and things like that. So this is the sort of two basic pools of genetic information a bacteria carries, the absolute necessities and the things that make them unique at the species level. And the sort of interesting thing about bacteria is, in addition to being a species, and we're going to talk about species in the next part of the lecture, but bacteria have different species, but sometimes bacteria have what are called different strains. And so to give you an example of what I mean by this, there's normal happy E. coli growing in your guts, but there are pathogenic strains of E. coli living that live in sort of in the wild, and they live in industrial settings that potentially can cause food poisoning. So the same species of bacteria, E. coli, but very, very different genomes that led them to live very different lives. One pathogenic, one symbiotic in your gut. So the bacterial chromosome is pretty small. As I mentioned, typically just one chromosome. Overall, it's about 1 millimeter in length. So it's pretty long. And this is long in the sense of, like, if you stretched it out and if you could physically see it, you could measure it on a ruler. 1 millimeter is a pretty long piece of DNA. Just to put this in perspective, the size of an E. coli cell is about 1 to 2 microns long. So the chromosome itself is 1,000 times longer than the cell. This is very much the same thing for your cells. You could take all the DNA in your cells and stretch it out, and it would be many, many thousand times larger than the actual size of your cells. Interestingly enough, the DNA of any bacteria, but in particular E. coli, does constitute about 2 to 3% of the total cell weight and occupies 10% of the cell's volume. So just to sort of, just to ring a bell, sort of remind you what this looks like, right? About 10% of this cell is this chromosome here. So bacterial genome size has pretty heavy variation base. I just mean, 2,000 AT Cs and Gs. That's all I mean. And so we can look at how the variability of genomes is. So we have some pretty big genomes over here. So we have Mycobacterium tuberculosis, Burkholderia cepacia. Both of these are pathogens. And then you can see some smaller ones. We have Borrelia burgdorferi here. It causes Lyme disease. And you can see some even smaller ones like this archaea here, which has 1,700 kilobases. So genome size varies pretty wildly, but you also notice that some bacteria have different number of chromosomes here. And you also notice that some bacteria have one or more plasmids. It's actually kind of interesting. The bacteria that causes Lyme disease, Burkholderia, Borrelia burgdorferi, I'm sorry, has 21 plasmids. So it's sort of a really unique organism. It has a small genome, but has many, many plasmids. And just as a note, genome size does vary considerably across the tree of life. And sort of an interesting thing you notice that viruses have the smallest genomes, followed by bacteria, fungi, and the biggest genomes on the planet belong to protists, plants, and some amphibians. So pretty substantial variation in genome size, right? We have the average bacterial genome is less than 10 megabases, and that we sort of scaled it up, right? The biggest genomes that we know of are 10,000 times the size of your average bacteria. And this is sort of an interesting phenomena here, is that, we sort of look at this and we see, well, mammals, aka us, which we typically think to be the most complex organisms on the planet, have, relatively middle of the pack genome sizes. And this is in contrast to protists like our Fusarium, which are single cell microscopic organisms. And they have some of the biggest genomes we know. And so there's this really interesting phenomena in eukaryotes that tells us that genome size itself actually does not correlate with organism complexity. Because we would imagine that if that happened, the things that had the biggest genomes, trees, mammals, would have much larger genomes than the very simple, such as protists. But this is direct contrast to bacteria. In bacteria and archaea, genome size correlates with metabolic flexibility. So the bigger your genome are, the more genes you carry, and the more things you can do. The more metabolisms you can have, the more antibiotic resistance genes you have, more antibiotic resistance, I'm sorry, the more antibiotic production genes you can have. So there's a lot of different things you can have by having a larger genome as a microbe, but those things don't really exist inside of our eukaryotic friends. Now, the genome structure of eukaryotes versus bacteria is very different as well. So just to orient you to this picture, green things are genes, yellow things are introns. So if you remember back from basic biology, introns are intervening sequences. So they're parts that break up eukaryotic genes. You'll notice that they're absent from our bacteria. We have pseudogenes, and pseudogenes are genes that have lost function. Human genomes have lots of pseudogenes, just as a note. And then we have this purple, which is what we call a genome-wide repeat. Bacteria is very different than eukarya, right? Bacteria is pretty much all green, whereas eukaryotes are all purple pretty much, right? Very, very different structure of the genes. And archaea actually have relatively similar genome structure to eukaryotes. They have introns, but they have a very, very high density of genes like we see in bacteria as well. They don't have a lot of this, what we would typically term junk DNA, things that don't actually encode for genes or proteins, right? This green region. And we could just sort of show you another comparison here where we have the amount of genetic material and repetitive DNA in various organisms. And what we can see here is humans, yeast, flies, corn, and bacteria. Anytime there's red, it's a gene. Anytime it's orange is a pseudogene or an intron. Anything that is purple here is a genome-wide repeat. And what you see is that our yeast and our bacteria are pretty densely packed with genes. This is direct contrast to humans and corn, right? A lot of repeats, introns, very, very different genetic structure. Now, flies are an interesting case here. They have a different structure than corn as well as humans. It's more similar to bacteria, but ultimately they still have lots of parts of their genome that actually don't encode for any proteins, so they're not actually genes. And so just as a final note before I move on, the density 70%. Something like this corn here would have a very low capacity at about 5 to 10%. So it varies between organisms. So next up on our train of DNA and genomes is plasmids, and these are extrachromosomal DNA. And so we have a bacteria here. It's been osmotically disrupted, so they essentially stuck it in salt water, and it exploded. And by exploding, it spilled all its genome out onto this agar. And what we're seeing here is actually its plasmid. And as I mentioned, plasmids are what we call accessory chromosomes. They're not part of the core genome of these bacteria, but they are accessory and they carry sort of interesting things. So we'll talk about that in a moment. So they are segments that actually replicate themselves autonomously. So they don't need the bacteria to do anything. They can do it all by themselves. They have no ribosomal genes, so they don't encode any ribosomes, and they have their replication is independent. They can actually have multiple copies in a cell. So this bacteria actually has two plasmids. You can see one here, and you can see one here. Some bacteria, as we saw with one of the examples in one of our previous charts, had upwards of 21 plasmids. So you can have many, many copies of these plasmids. Now, when we look at how many bacteria, naturally speaking, have plasmids, the copy number seems to be low. Most bacteria in nature, and this includes your own gut or your skin, typically have one or no plasmids. But in the laboratory setting, and as well as the clinical setting, the amount of plasmids per genome, again, can be pretty high. And so there's actually a really interesting application we can use our plasmids for, and this is a technique called cloning. And so the way this works is we have a cloning vector or a plasmid. We break it open to get sticky ends. And what we can do is extract a gene from a chromosome of interest. It can be my genome, it could be from your genome, it can be from another bacteria's genome, it could be from a fungal genome, it could be from any genome. But we can take a piece of a DNA out of any genome, tack on some sticky ends, and have it merge with a plasmid. And what's sort of interesting is we end up getting a plasmid that has a gene of interest on a plasmid. And what we can then do is use a process called transformation, which we'll talk about in a few classes, and stick it inside of a bacteria. And the interesting thing is, once it's inside of a host, the bacteria will naturally express that gene, whatever you stuck in that plasmid. And so this process of cloning is a useful vesicle for the acquisition of new traits in nature. But we've used it, industrially speaking, to make very important molecules such as insulin. So I'm just going to go back to this one slide right here. Using this process of cloning, we've stuck the gene for insulin inside of a plasmid, then stuck that plasmid inside of E. coli, and we've been using E. coli for a couple decades now to make synthetic insulin for diabetics, which is actually a really, really important thing for diabetics because I don't know if you guys have ever heard, historically speaking, how diabetics used to get insulin, but they used to get it from the pancreas of pigs. And one of the sort of the things about when you're extracting biologically important compounds from animals is that they vary in strength. So sometimes you get a really strong dose of insulin. Sometimes you get a really weak dose of insulin. You never really would really know. And it was always a really dangerous thing. And the people that had to use insulin were sort of taking their life in their hands every time they injected this really important life-saving drug. But using cloning, using this application, we actually can use it to make consistently strong insulin so diabetics always know how much insulin they need to give themselves. So it's a really important thing. Now, naturally speaking, it's a great way to acquire new traits. And so, for example, here, a soil bacteria could potentially acquire a plasmid that will give it new genes to utilize some potentially new nutrient source. We can also see in a clinical setting stuff. And so plasmids that carry non-beneficial things can also persist in the populations as well. So that's bacterial genome structure. Now I'd like to move on to the next topic, which is going to be bacterial differentiation. Now, when we talked about how bacteria divide, daughter cells are typically genetically identical to their parent cells, and they don't have to be, but they don't have to be physically identical. And what do I mean by that? Well, pretend you have an identical twin. You have the same genome, but pretend one has red hair and the other one has blonde hair. That's what we're talking about for these bacteria. And so cells can actually differentiate depending on the environmental conditions or the life cycle of an organism. So I'd like to talk about this. And the first lens we're going to look at this through is actually through a really interesting bacteria called Caulobacter crescentus. Um, Caulobacter crescentus is a waterborne pathogen. It lives naturally in fresh water, but it can cause infections in humans when we're exposed to it. Every time Caulobacter crescentus divides, it produces two daughter cells like all bacteria do, but each of them has a distinct cellular fate. One of the daughter cells is a stalked cell, and the other one is a swarmer cell. And these stalked cells are sessile, and these swarmers, as their name suggests, they're motile. They have the capacity for movement. But the interesting thing is, once the swarmer finds a good place to attach, it transitions into a stalk cell. So let's look at what's going on here. We have free swimming Caulobacter crescentus. It's swimming, swimming, swimming. It finds a place to hold on. It extends this holdfast stalk here. Once it extends the stalk, it grows it a little bit, and then it starts to divide. Because remember, that's what all bacteria really want to do. The stalk cell divides, producing a swarmer cell, and that swarmer cell goes off to then find its own place to stick and attach to and repeat the cycle over and over again. Another really important, especially in a clinical setting, is bacterial differentiation with respect to endospore formation. And endospores are a really cool thing in my book. I find them really, really fascinating. Now, what the heck is an endospore? Well, as we talked about, bacteria have to respond very rapidly to the environment. And the environment doesn't always have to be good, right? They don't have to respond in a positive way to the environment. Sometimes the environment is awful, right? Maybe there's not enough food, there's not enough water, the temperature's too hot, or the temperature's too cold, or there's just too many different types of bacteria around, or maybe there's antibiotics around. What endospores do is this special type of cell that produces besides something that it's really, really harsh, like gamma radiation, an endospore can protect these bacteria against. And just as a note, as a biological entity, um, endospores are known to be the most biologically hardy um, structure on the planet. They can survive anything. You could take them out into space. You can put them above the boiling point of water. They can really survive anything. Now, let's talk about some of the sort of the way these microorganisms grow, how they sporulate. So, normal vegetative growth looks like this, right? One cell splits, becomes two cells. Pretty straightforward. Now, let's look what happens in our overall step of sporulation. So, the first thing that occurs is a septum forms. It forms near one pole here. The DNA replicates itself and then extends to fill this sort of extra part here. Step two, the septum does separate and the spore itself begins to form. And it forms inside the original mother cell. But what you'll notice is that it formed again directly inside, but it's still at this point at stage 3 here. It's still attached. By sort of the later stage 3, the mother cell does engulf what we call the forespore, surrounding it with a second membrane. So you can see sort of it extending out, trying to cover it again. Once engulfment occurs, the chromosomes of the mother cell begin to disintegrate, eventually disappearing even more, and then disappearing even more. And the most important step here is the formation of the exosporangium. Now, what is that? So what ends up happening is this forespore here develops a coat of peptidoglycan between the original forespore membrane and the membrane from the mother cell. And this is basically a coat, a really hardy coat of our friend peptidoglycan. It also coats it with protein as well, which gives it its overall spore coat. The final step here is we synthesize diapoclonic acid, and calcium is incorporated into the spore coat. So you get this really hardy structure that's a combination of protein, it's a combination of sugar, it's a combination of calcium and acids holding it overall together. And one of two things happens with the spore. Now, the spore can stay inside the mother cell, so that's one type, or the mother cell can break open and releasing the spore into the environment. It just really depends. But once the spore is formed, as I mentioned, it's one of the hardiest biological structures on the planet. And these cells are really interesting. They can be very highly resistant and metabolically dormant spores can remain in viability for thousands of years. So, for instance, every time they do archaeological studies, they can actually cultivate historic bacteria from thousands of years ago. And I mentioned that you could take these spores up into space. They're wild and wacky. Now, you can look at this is a microscopic view of our spores, and you can see that some spores are still inside them other cells, some have been released. It depends on the species of bacteria. Some always release, some never release, some release depending differently on the conditions. It just really varies. So in terms of how this works, it's if once a bacteria sort of encounters a nice happy situation, essentially the spores actually germinate very much like a seed does. And it begins only when nutrients are available. And during germination, the endospore actually just simply picks up where it left off. It dissolves its coats around it, starts to form a brand new membrane, and starts be doing it again. And then it starts essentially picking up where it left off before it began sporulating. For clinical science and for doctors and nurses and anyone who's involved in this is that they're actually really good as a sort of way to transmit disease. And what do I mean by this? Well, for those of you that have ever heard of, you know, um, you know, swollen soup cans. So swollen soup cans are one of the ways you get Clostridium botulinum, which is botulism poisoning. What's happening is that there were spores inside those cans. They germinated, um, causing Clostridium botulinum to grow and then potentially be infectious. And so spores are a really powerful way to transmit diseases, and there are many, many clinically important bacteria that use spores as a means of being more infectious, as a means of surviving antibiotics, as a means of being transmitted from host to host. And these include such nasty bacteria such as anthrax, tetanus, botulism, as well as gas gangrene. As a note, just as on a more fun side of this sort of sad topic, is that Clostridium perfringens is a really interesting microorganism. When it does infect epithelial cells in individuals, it actually produces a compound that smells like grapes. So if you have someone that has gas gangrene, you can actually cut them open, and it actually smells like grapes. So just as a fun note here. But for this very reason, because they are so involved with some pretty serious diseases, if you get anthrax, you get tetanus, you get botulism, you have a very, very high probability of dying. Understanding the function of spore coats is really important to preventing these infections and diseases. So, for instance, tetanus is really difficult to treat, but we have a vaccine that targets the toxin of tetanus, allowing us to recognize it. The same thing for many, many other spore-forming diseases. But ultimately, are very highly resistant to heat, UV radiation, and environmental toxin. The exosporium is actually a pretty poorly understood layer of protein. The spore coat itself is a very dense protein layer that consists of at least, if not more, 70 proteins and provides resistance to chemical and enzymatic treatments. So, for example, there's many, many proteins out there in this that are involved with detoxivation of oxygen-free radicals, which is a pretty common thing that your white blood cells will expose the bacteria to when they engulf them. The cortex itself is a very thick layer of peptidoglycan. We've already talked about that pretty um, in depth in the cell wall lecture. But the endospores themselves are highly desiccated, and this helps them protect them from chemical damage during dormancy. So it's a really sort of a nasty sort of overall thing. In addition, the DNA of a endospore inside of an endospore is coated with a specialized protein called SASPs, or small acid soluble proteins. These protect it from damage by altering the physical structure of the DNA. There's also calcium or dipoclonate. It's something that's abundant within the endospore, and it seems to stabilize and protect the DNA, but we're not entirely sure what it does. But again, it seems to act in stabilization of the DNA. And there's also DNA repair enzymes. And what these DNA repair enzymes do is if something gets through all this and damages the DNA, these enzymes pick up the slack and fix the problems that any damage that got through caused. And if we were if we were in person, and we'll do this sort of virtually, we would do some spore stains, and you can look at some cultures of bacteria here. The bacteria, the sort of the mother cell is stained red, and the spores are stained green. And then you can also see a picture where the spores are outside the cell. It's again, it's a feature lab activity. It's not really fun to do. It almost never works when we do it in person. So I'm actually kind of glad we're doing it remotely this semester, even though I'm not particularly happy that we're not doing in-person labs. But I digress. So next up on our topic of bacterial differentiation is going to be heterocyst formation, and these occur in cyanobacteria. And these are really interesting organisms. So if you sort of remember back to our we sort of talked about how different types of bacteria in one of the early classes, one of the types we talked about was cyanobacteria, and these are single cellular organisms, but they form these chains. So this is one cyanobacteria, this is a second cyanobacteria, but they form these links. They're kind of like you can think of it like a sausage link if you so choose. So they form these filaments, but they have these very specialized areas called heterocysts. And these heterocysts are specialized locations for nitrogen fixation. And what the heck is nitrogen fixation? Well, nitrogen fixation is where a bacteria take nitrogen that's available in the atmosphere and convert that nitrogen into ammonia. And ammonia is, basically fertilizer, right? That's what we spray on crops. Now, the interesting thing about nitrogen fixation is that it occurs irreversibly inactivated. And these cyanobacteria, they are photosynthetic as well. So they generate their energy through photosynthesis, and they produce oxygen. And so the sort of interesting thing is we can sort of break down this reaction. So we have nitrogen from the atmosphere. It's converted into, it's essentially you tag on some hydrogens here. So it's NH, NH, breaking the one of the three bonds in the triple bond. And then that is then converted to two NHs. And then at the end of the day, you convert it to NH3, and that's ammonia. So that's what you use to make your amino acids. That's nitrogen. And the overall reaction requires lots of hydrogen, lots of ATP overall. So it's a pretty energy intensive process. But the important thing about this process is that the heterocyst, and this is where the heterocyst comes in, is that nitrogen fixation is catalyzed by an enzyme called nitrogenase. And nitrogenase is again what's doing all this chemistry. Now, the interesting thing about nitrogenase it is that is it's super susceptible to oxygen. And once nitrogenase experiences any oxygen, even a tiny, tiny amount, it becomes irreversibly inactivated. So you need an anaerobic or an oxygen-free environment. And you might think to yourself, well, how does a photosynthetic organism, which is generating oxygen all the time, how does it physically keep its nitrogenase safe from oxygen? Well, it does this by compartmentalizing its two reactions. Photosynthesis occurs in the regular sort of filamentous zones of this photosynthetic organism, and nitrogen fixation occurs in these heterocysts. And so just as a note, this heterocyst, before we sort of wrap up this section, our night is completely oxygen free. Oxygen doesn't penetrate. It's got a really specialized coat around it that prevents any oxygen from coming in. So it's a really interesting case of how you can specialize an organism, um, grow them together, again in this chain, to accomplish one overall goal. And so overall, though unicellular, some microbes can differentiate to suit their environmental or metabolic needs. Just as a note, this won't be the last time that we are seeing microbial cells acting like cells from higher organisms, right? This specialization is very much like the human body, right? The liver cells do detoxification, right? The heart pumps blood, right? Specialization at the cellular level. So this is something we do see in microscopic organisms. So just to wrap up what we learned in this very first part of the lecture. So first is that prokaryotes have varied genomes with different size, shapes, and numbers of chromosomes, as well as a different number of plasmids. Prokaryote genomes themselves contain core and flexible elements, and these flexible elements encode all sorts of wacky and wild things like antibiotic resistant genes, um, antibiotic production genes, cool genes for detoxification of heavy metals like mercury, all sorts of crazy things. Um, in addition to specializations and changes in their genomes, bacteria have the capacity to differentiate depending on certain conditions. And the sort of the most important thing we talked about is actually spores, and they are very, very hardy. They're designed to survive long-term in very harsh conditions and play a pretty large role in some pretty deadly pathogens. So that's going to be part one. That's just introducing the genome. That's introducing how bacteria can differentiate. So let's dive a little bit deeper into this idea of the genome. How do we replicate a genome? And so we're going to answer a couple questions here, but we're going to take a quick detour before we do that. So the first question we're going to answer is, what the heck is a microbial species? That's going to be our detour. Second is, how do bacteria replicate their genomes, and what are the key proteins and enzymes involved? What are the steps of genome replication? And how do plasmids and archaea replicate their DNA? And how does it differ from bacterial core genome replication? So what is a species? It's kind of a difficult question to answer for microscopic organisms. Now, when we think about biology as a science, we need this fundamental unit to describe different organisms, right? Humans are a species, dogs are a species, cats are a species. The way we typically think of how to differentiate different species of a macroscopic organism is the capacity to mate and produce a viable offspring. And this typically does require sexual reproduction, but I will say one thing. Viable offspring means that your offspring can then go on to produce their own progeny. So something like a mule, which is a combination of a horse and donkey, is not viable. It's completely sterile. So it can never produce babies. So thus, a donkey and a mule, even though they can mate, are distinct species. And so this typical definition does not apply to microbes, because microbes don't mate, right? They don't reproduce sexually. They reproduce via binary fission. So this species level definition that we use for macroscopic organisms doesn't quite apply to our microbial friends. So as biologists, we do need a way to classify organisms. Everything needs to be classified, and it's not just just because humans like to classify things, because we really do. That's like one of the most basic things humans like to do. But we needed a way to be able to talk about different organisms. And it's useful for taxonomic units for many aspects of biology. So not only for differentiating organisms, but also to understand evolutionary biology. And it's also very useful for measuring diversity. How many species do I have in area X? For microbiologists, we have this problem. How do we define a species? There's no sex, right? It's all binary fission. Well, there's two possible solutions here. We can use what's called functional relatedness, and this is um, the way we used to view things um, prior to um, prior to sort of the DNA revolution in the 90s. The idea being here is that you could differentiate between two species depending on what functions they possess. Same pathways. And that's what we call convergent evolution. In addition, there's also lots of redundancy in microbial function. And so you can have one enzyme that does multiple things. And this is common in very distantly related organisms. You can have two bacteria that are separated by millions of years of evolution, but that can do the exact same thing. And then finally, the most pressing problem here is actually this concept of horizontal gene transfer. And we're going to talk about this more in depth in a couple classes, but just as a note, horizontal gene transfer is when bacteria, as well as archaea, can acquire new functions, either through taking DNA pieces directly up from the environment. This is a process called transformation. They can get new DNA from viruses, or they can get DNA directly from conjugation, which is a process by which bacteria swap plasmids. And so bacteria can do this again from the environment, from viruses, or swapping DNA of the bacteria to acquire new traits. And it doesn't have to be something that's closely related to them. You could be something that is very, very distantly related. And just to sort of show you what bacterial conjugation looks like, we have two cells. One is what we call an F positive cell, which means it's ready to perform conjugation. And we have an F negative cell, which is not quite ready. But this F positive cell extends the sex pilus, changes this F minus cell. And again, we'll talk about this in a few classes, to then swap genetic material. The second possible solution is genetic relatedness. And so we can look at how similar a genome of a bacteria is, or use the similarity of very, very conserved genes. Genes that change very little over evolutionary time and are present across all bacterial species. And the genes that are sort of the best candidates for this are actually what we call ribosomal genes, so genes that encode parts of the ribosome. As we mentioned, all cells have ribosomes. And so ribosomes are super important to how bacteria function, so they evolve very slowly. And so they're actually very useful as a biomarker because they evolve slowly. And so things that are closely related actually have more similar ribosomal. And they share 97 bases in common. You would consider that the same species or the same operational taxonomic unit. And in this scenario again, if we had again 100 DNA bases and 96 of those 100 were similar, we would consider those a different species or a different operational taxonomic unit. And so this is a pretty good way, just comparing genetic relatedness, but there is a problem with this. Um, um, it ignores function. And so there's a really big issue in the medical world of comparing E. coli Shigella problem. But the best solution ultimately to define a bacterial species would be to combine function and genetics. Getting genetic similarity is easy. Getting function can be hard. You have to get function of a bacteria, you have to culture a bacteria. And that's really hard for a lot of organisms. Many organisms are not culturable on plates. In fact, when you look at when we try to culture all the organisms from any sample, whether it's your skin, whether it's a soil, less than 1% of the bacteria we can actually physically grow in culture. So it's very hard to determine function of bacteria. So the way to define a bacterial species, we're not quite there yet. There's much debate about what's the best way to go about this, what's the best cutoff to use for the genetics, how do we culture things. So for now, when we're discussing a bacterial species, we typically call it a taxa or a taxon or an OTU or an operational taxonomic unit. But we'll use species from time in this class. And we can use species not only mostly because we're talking about very well-known and well-studied organisms. But if you start to look at say when we dive into the human microbiome, species is not really appropriate to discuss what lives on and inside a human because we just don't know enough about them functionally and genetically to make that discussion point. So with that out of the way, let's move on to DNA replication. And so DNA replication is something all The DNA is replicated bidirectionally, so it goes left and right from this replication fork, and the replication ends at the terminus, um, which is going to be down here. So this is what we're talking about here. Now, just as a note, when I say terminus down here, the enzymes that do this synthesis here meet and then stop. Bottom strand of DNA. And so what we get here is when we're copying this double-stranded DNA here, we have these replication forks, which is the splitting of the DNA. And what you're seeing here is the parent strand here is in gray, and the new piece of DNA that we're synthesizing is here in purple. So when we're replicating a genome, we create, again, two identical pieces of DNA. But each of those two identical pieces of DNA contains half of the DNA from the original parental DNA, and half the DNA is from the newly synthesized strand. So this is what we mean by semi-conservative, is that half is from new stuff, half is from old stuff or the mother's DNA. Now, DNA replication is catalyzed by DNA polymerase. So just as a note, anytime in any biology course you see any word ending with ASE, this is an enzyme. Now, an enzyme is as a protein that does chemistry. But anytime you see ASE, we're always talking about an enzyme. Now, replication of DNA is done and catalyzed by DNA polymerase, and this is done within what we call a replosome. Now, a replosome is a complex of proteins. It contains DNA polymerase, helicase, gyrase, primase, and ligase. And you can sort of see all these things over here in this figure on the right. And each DNA replication, each DNA replosome has two DNA polymerase. One is for the leading strand and one is for the lagging strand. And so what do I mean by the leading strand versus the lagging strand? Well, if you remember back from early on in your first science class here at Roxbury, DNA has directionality. It has a 5' end and a 3' end. DNA polymerase can only read from 5' to 3', like a book, right? We only here in the United States, and those that speak English um, only read from right to left, right? And that's the same thing here. DNA polymerase can only read DNA from 5' to 3'. So the leading strand, DNA polymerase is reading in the proper direction. It's going 5' to 3', so it's just chugging along. The lagging strand is the opposite, right? It's 3' to 5'. So the DNA polymerase actually has to jump back and synthesize, because remember, it can only go from 5' to 3'. So what ends up happening is it synthesizes a little bit this way, jumps back, synthesizes again, jumps back, synthesizes again. This is why we call it the lagging strand. It just doesn't move as fast as the leading strand. So let's talk about some of this organization. So the origin itself, when a bacteria is getting ready to divide, it moves to the cell's pole. So we can see the origin of replication here, and we can see the two cell replisomes here forming. Now, the replisomes, in this case, it's yellow. They stay in the center of the cell during replication. So these replisomes don't actually move. They just stay with the origin of replication. They stay at the pole of the cell. And the terminator, so the part where we decide where the replosome knows to stop synthesizing new DNA, actually stays in the center. You can actually physically look at this. We have the origin of replication, the terminator, right? This is what it looks like at the cellular level. Now, in this example, it's moving to the poles, upwards. It can also move to the side completely. It just depends on the bacteria. So let's dive a little deeper though. Let's start with some of these key enzymes that we want to know. And so genes, there are many, many genes that are involved here. So one of the important genes is the origin of replication. It's called OREC. It's a very small gene at 245 base pairs in length. Next, we have these DNA-based ones, and these are involved with initiation and binding. They're helicases and they're loading factors. We have polymerases, primases, and all the genes here. So this is all the genes that we know of that are involved in DNA replication. There's a lot. It's a pretty complex group of machinery, and you'll see very shortly what this looks like. So as we mentioned, DNA replication is bidirectional. And so we have the origin and the terminus here. For DNA replication to start, we actually have to form a bubble. And this creates two replication forks. So we start here, origin, terminus. We have a bubble forming at the, the, um, the origin, and then our replisomes move bidirectionally. So they go left and they go right here. And you can see the enzymes are they're going. They're synthesizing as they're going. They have this overall fork. And eventually they reach the terminus, forming two distinct bacterial chromosomes. So that's the workflow we're thinking about here. So let's talk about this. So first, we start replication, as I said, at the origin or ORIC. Now, our enzyme DNA-A recognizes and binds to a 9 base pair repeats at the origin. Sort of as an interesting thing about DNA-A, as a cell grows, so bacteria, before they divide, they will grow. It actually starts to build up DNA-A inside of its cell, getting ready to divide. Once a sufficient amount of it is bound, DNA then begins to fold and creates an opening. So that's what DNA-A does. It causes this, it binds to this 9 base pair thing, it causes this slight opening, and then things really get started. The next enzyme in our process is DNA helicase, or DNA-B. It attaches to this opening, so you can see it um, DNA-B as well as DNA-C, which is a helicase loader. Both of these things bind. So just as a note, the DNA has to the DNA-B has to recognize DNA-C to bind. And what it does is it actually recognizes a 13 base pair strand near the origin of replication. And these two bind to the DNA. And eventually DNA-C is lost, just leaving DNA-B attached. And then the DNA itself begins to unwind. And this is when things start to get involved. Now, as it does unwind, the small single-stranded DNA binding proteins, which we call SSBs, actually coat the exposed single-stranded DNA, protecting it. So you can sort of see these here, these sort of right down here and over here. It essentially starts to coat the protein, the DNA, protecting it, because you want to, this requires energy, and anytime something requires energy, you could potentially damage your DNA. So you have to protect it. But the next step in the process is that DNA helicase recruits DNA primase. And what the heck is primase? Well, primase is important. So as we mentioned, DNA polymerase is the enzyme that catalyzes the synthesis of new DNA strands. And for DNA polymerase to work, it needs the 3' end to start building onto. It needs what we call a primer. And for a primer to be formed, we need the DNA enzyme aptly named primase, or DNAG. This is an enzyme called an RNA polymerase, and what this does is it creates a primer, again, that recognition zone made out of RNA. And this RNA primer has what is, just as a note, has a 3-hydroxyl group that DNA polymerase itself recognizes. So we see our helicase here, a primase comes in, tags it with RNA, letting DNA polymerase know, hey, come bind here. Now, the other thing that our primer does, it actually recruits a multi-subunit complex, which is off. It's essentially, you can think about like a guide protein. So what does this look like? Well, it's got this sort of odd structure. It kind of looks like an alien. Like this is the alien's head, and these are the sort of antennas. And the antennas are where DNA polymerase binds to, and the sliding clamp essentially what essentially snips on to the DNA strand, guiding the polymerase. But what it does is that once this complex is bound, it positions the DNA polymerase on the primer itself. And once it recognizes that primer, it then can be synthesizing new strands of DNA. Now, um, as I mentioned, the RNA primer itself has a 3-hydroxyl chain, and DNA polymerase actually only proceeds in a 5' to 3' direction. So um, which is easy when you think about how this works, right? So if we have the leading strand down here, the DNA polymerase is moving from the 5' to the 3' region. And so that's pretty easy. But the lagging strand, remember, it can't go 3' to 5'. It has to go 5' to 3'. So what you end up getting is the lagging strand. It sort of has interrupted synthesis of new DNA, whereas the leading strand has uninterrupted DNA synthesis. In terms of the lagging strand synthesis, let's talk about that a little bit more. So the DNA polymerase itself does continue up to its previous primer. And the idea being is if we have this replication fork, this is we have our leading strand here and our lagging strand here. What ends up happening is, as I mentioned, the DNA polymerase has to go 5' to 3'. So what ends up happening is um, the DNA polymerase will essentially jump back to its primer, which is going to be this green area, synthesize this way towards the 5' end, jump back, synthesize again recognizing a primer, jump back, synthesize again, jump back, synthesize again. And it does this not in very short pieces, and it does this in these 1,000 base pair pieces, which are called Okazaki fragments. You might have remember this from your intro bio class. This is a Japanese individual who actually won a Nobel Prize for this. It was a really important discovery at the time. But this is what happens in the lagging strand synthesis. You have all these strands, which again is in direct contrast to the leading strand, which remember, these are lagging strands, so it disconnects from, once it gets that 1,000 base pair strand done, it disconnects, finds a new primer, and then synthesizes the next 1,000 base pair strand. And does this over and over and over and over again. But the important thing here, you should also note is that there's lots of SSBs. So these are small DNA binding proteins that help protect them. And so it does protect the strand as it's sort of doing this lagging strand synthesis. Because again, anytime something requires energy, you can potentially be done. Now, ultimately, whether you're on the leading or lagging strand, both strands move to the termination site. Remember, this is the area where termination stops. The polymerase recognizes this strand of DNA, says, hey, I'm done, and it stops synthesizing new. Now, remember, this happens simultaneously, but it basically happens as like a circle. They sort of go around the circle in opposite directions, clockwise or counterclockwise, until they meet in the middle. Um, And remember, our replisomes themselves are actually stationary, so the DNA actually kind of gets fed through the replisomes. But when the replisomes meet at the terminus, they collide and then ultimately disconnect from the DNA. Now, the important thing here is that our leading strand is one solid strand of DNA. But remember, our lagging strand is multiple pieces of Okazaki fragments, right? About 1,000 base pairs. So what needs to happen is that for our lagging strands, they have the RNA primers that we've been adding along to start new synthesis of Okazaki fragments have to be degraded, and they're degraded by an enzyme called RNase. Any gaps that are present in these Okazaki fragments are then filled in by DNA polymerase, and then an enzyme called ligase essentially nicks all these DNA bases together. So eventually stringing all the Okazaki fragments together. Pretty straightforward process, but this is an important thing, not just in bacteria. This is almost identical to what happens in humans as well, just as a note. So that's typically what we think about when we think bacterial replication. So let's talk a little bit about the other two types of replication, which include archaea and plasmid. The question you should be asking yourself: should it be similar to bacteria, or is it similar to eukaryotes? It's actually more similar to eukaryotes, but in respect to the enzymes and the structure of the enzymes. But the thing about DNA replication is it's very, very old. The enzymes present, while they vary between bacteria, archaea, and eukaryotes, it was evolved very, very early on in the tree of life. And so DNA replication behaves very, very similarly across all three groups. But the proteins and enzymes do vary. And the archaeal machinery itself is actually much more like eukaryotes. but archaeal genomes, interestingly enough, are actually much more like bacteria. And this is just a nice table showing you all the different names of the genes of bacteria, eukaryotes, and archaea. And for archaea, we have two groups, the Eukaryotic and the Crenarchaeota. And you can see the chromosome, the origin, replication, origin, recognition, and all the different enzymes we just talked about, and all their different names here. But again, similar functions, just slightly different genetic material, and some additional genes in our eukaryotes and archaea relative to our bacteria. So let's next talk about plasmid DNA replication. So replication of many of these extrachromosomal elements is not tied to the replication of the chromosome itself. It is autonomous. And so each plasmid contains its own origin sequence for DNA replication, but there's actually very few of the genes needed for replication itself. And so many of the proteins, when a plasmid is replicating itself, are actually the host's own enzyme. And this is actually a sort of interesting thing. Uh, Viral genomes are actually structured very, very similarly to plasmid genomes. So some people think that viruses originated as plasmids, but that's a topic for a whole nother day because that's a whole lot of speculation. um, so our plasmids replicate in two different ways. They can do bidirectional replication, like a bacterial chromosome, which occurs in the two directions, or they can do what we call the rolling circle replication, which is unidirectional. So let's talk about what those two look like. We can do, again, the bidirectional, um, very much like the bacterial chromosome, or we can think about it as a unidirectional where a replication begins at a fixed point, again, the origin, but only moves in one direction. And so as the polymerase moves, it actually does displace one strand as it replicates the other. So what do I mean by that? It basically just strand of it separately. So just sort of a weird way of overall doing that. So let's just sort of move to the end of this lecture with a little bit of fun math. So how fast does an E. coli polymerase synthesize DNA and nucleotides? Well, the genome of E. coli is 4.6 million bases, and replication is bidirectional. So the chromosome completes the replication entirely in 40 minutes, which if you do the math behind that, it means you has a synthesis rate of 1,000 nucleotides per second. So that's really, really fast, right? Just imagine like trying to grab a thousand, uh, trying to grab like a thousand, like I don't know, like bubbles at a time, and that's how fast DNA replication is occurring. So just to summarize this part, a species is a concept still not solved for microbiology, but you'll typically see, and as you sort of do your readings for your microbe wiki, you'll see taxa being used instead of species. Bacterial genomes replicate bidirectionally and begin at the origin of replication and extend all the way through at the terminus. And this has two replication forks. Each one has a replosome that coordinates and recruits proteins that mediate DNA synthesis. And archaea, as I mentioned, are more similar to eukaryotes in respect to the enzymes, but they do replicate their DNA very much like bacteria do. So with that, that will be the end of today's lecture. I do hope you learned something. And please, as always, if you have any questions about what we um, what we covered in today's class, if you have any questions about due dates, if you have any questions about assignments, please just let me know. I'm here for you. I can talk to you whenever you need me. So as I mentioned, with that, I will end today's lecture, and I hope you guys take care. Goodbye.